How was the quiz? Was it easy?
Difficult.
Difficult? Okay, what part was difficult? Be honest. Everything.
Everything.
Okay, so my idea is to put very simple questions like what is anatomy, what is physiology. At least you should know that because you're taking this course. Some other questions may be difficult, like the one on homeostasis perhaps, or to mention the phases of mitosis. Very quickly, anatomy, just a refreshment. Study the structure and shape, the body and its parts in relationship. Physiology, it's how they work. Okay, how they work. One is the structure, the other how it works, the function. Basically, you will have to describe this graph. It's, I think, in the first or the second session that we have. Remember, we receive the stimulus, the receptor receives the stimulus, sent through the afferent pathway to a control center. The control center will integrate and decide what action to take. We'll send information through that afferent pathway to an effector, and then we will have a response. Basically, what we need is to keep within normal ranges, right? That is homeostasis. Then we can have negative feedback or positive feedback. So review this part of the class so that you refresh your memory. Okay? About the cavities.
This is most difficult.
Oh, really? I thought this was going to be one of the easy ones. Okay, so we have the dorsal cavity, ventral cavities. In the dorsal one, we have the cranial cavity, which contains the brain. We have the spinal cavity containing the spinal cord. The ventral cavities, we have the thoracic cavity. Basically, we have the lungs, the heart. We have the abdominal pelvic. Sometimes we can divide it, even though there is no physical division among them. Abdominal cavity will house your stomach, your liver, your intestines. In the pelvic cavity, lower part, you will have your reproductive organs, your bladder, your rectum. You could very well just mention only one, the abdominal pelvic cavity, and mention all those organs, just to save you time and to think a little less. The question was to mention, right? Mention the stages. So whenever you're asked to mention, it's enough to say prophase, metaphase, anaphase, and telophase. That is pretty much just mention.
Okay.
If I say explain or describe, then you'll have to really explain, okay, at this phase, this happens, the chromatin, the chromosome, blah, blah, blah. They will separate, move towards. But in this case, I was just asking you to mention them. So if you only put these 4 words, it's okay.
Okay.
If you didn't remember these words and you wrote, sometimes the students make the drawing. Okay, I don't remember the names, but I remember more or less what happened these, the chromatins, they separate. I consider that okay. I'm not square-minded, you need to know the name, because it makes no sense to know the name if you actually don't know what happens. So in my case, usually I consider both okay. This is what I'm asking. So if you give me this, it's okay. If you don't remember this, put whatever you remember, okay, just to be safe. So you can draw, you can explain, it will be okay. This is for future advice, suggestions for exams. So maybe now it was not so bad. You will have better grades. Okay, basically I will try to cover the classes in the quizzes. Try not to be late so that you don't lose few minutes when you come. Quizzes are unannounced, so unfortunately you have to come. Yes.
Quizzes will be back. Can I receive
If I give you back,
Ah.
usually I don't give them back
Ah.
because there are only 4 questions, but I keep grading them. If you feel your score is not going well throughout the course, we can always try to figure out another way for you to raise your score. But the point is for you to attend, study, and get something, not just come and sit. Last year, I only have 1 withdrawal from class, so the student probably along the way felt it was too much. And in that case, if you don't want to continue, there is a period of time that you have to withdraw, okay? Formally withdraw from the course. Otherwise, I have to fail you, and you will have that in your record, okay? So in case I forgot, I already said you have to officially withdraw, okay? I try not to make it too difficult, okay? If you think it's too difficult, let me know. Then we will work it out, maybe explain it better, put more time into certain topics. So today I want to talk about the nervous system. The nervous system is very extensive and there's a lot of things to memorize and to do. Most probably we will not finish today because it's already more than 1:30. What is the main function of the nervous system? What would you say? Any ideas? What do you think it's in charge of?
Control our body.
Yes, to control our body, basically. It will control our body, it will communicate also different parts of the body, okay? But the thing is, the nervous system will make this communication rather faster than, for example, the endocrine system. Endocrine system also communicates, uses hormones, right, to get responses from your organs, the same way as the nervous system. But the nervous system uses a different pathways, and it's much faster than endocrine system. So basically, functions will be monitor the changes or the stimuli that are happening inside our body and outside our body. Okay. This system will process and will you forgot. Here. In case you didn't come last week, this is from last week. It will process and interpret all the information that is collected, which is called the sensory input, and make some decisions. What should we do, or what should certain organs should do? So then it will cause a response by activating muscles or glands, right? So I will either move, or my glands, I will start sweating maybe, or become more oil, or other types of glands maybe secreting. So does this process reminds you for our figure of homeostasis? We have a receptor, we have this pathway, we have a center of control, and then we have an organ that is doing something. Okay, all these pathways. Here comes the information as a sensory input. It's integrated in the nervous system, and we have an output. And how is it organized? So, by structure, we can make big distinctions: the central nervous system and the peripheral nervous system. What would be the main difference? One is central and the other one is peripheral. Okay, remember this. And by central we mean the brain and the spinal cord. Okay, so we have our brain and our spinal cord. This is the central nervous system. The nerves that comes out will be the peripheral nervous system. Okay, in the peripheral nervous system we have different types of nerves. We have the spinal nerves and the cranial nerves. Spinal or cranial, depending at which level they are carrying the impulses to and from the spinal cord or to and from the brain. Okay? Cranial from the brain, from the cranium. Okay? And if we see the function, what is the function? Then we can classify them as sensory or motor division. Okay, the sensory will be the afferent division. Remember afferent from the figure, something that is coming into. This is the somatic sensory fibers, or we can have visceral sensory fibers. So we are receiving information from these places. And the motor or efferent divisions could be somatic, or in other words, voluntary or involuntary, somatic nervous system or autonomic nervous system. Here we have in the figure, we have our central nervous system, brain, spinal cord, and we have the peripheral nervous system. Okay, we have the sensory or the afferent pathway, the afferent division. Comes here to the central nervous system, it's integrated, decide what to do, and information comes here through the efferent. Could be somatic or voluntary. Voluntary, basically those innervating muscles so that I can move, okay? I decide to move them. Or they can be involuntary. Could be sympathetic or parasympathetic. These two are basically, they have opposite effects. For example, one can raise your heartbeat, the other one can lower your heartbeat. Yes.
Are spinal nerves sensory division, cranial nerves motor division?
We will see more about that.
Okay.
We saw this slide when we were talking about the main body tissues. Remember the epithelial, the connective, the muscle, and the nervous system. So the nervous tissue is composed of the cell, called in this case neurons. Right? There are different types of neurons. Basically, they all will receive and conduct impulses, electrochemical impulses, and we will see why it's electrochemical. But the major function of the neurons are two: is irritability and conductivity. Okay, so that you don't forget and take note, because I printed the handouts before I edited the slides. Irritability is the ability to respond to the stimulus and convert it into an impulse. Okay, this is irritability. And conductivity is the ability to transmit the impulse either to another neuron, to a muscle, or to a gland. So these are major functions of the neurons. Okay? And remember that tissues, usually we have the cells and the supporting cells. In this case, the supporting cell in the nervous system is called neuroglia. What these type of cells do is insulate, support, and protect the neurons. Okay, neurons are extremely important so that all your body and your higher functions can properly work. Probably you have studied the general idea of neurons at school, perhaps? Do you study this at school? Yes? Yeah. Long time ago, probably. Yeah. Well, this is just a review. So the supporting cells, the glia, in the, what was CNS? Yes, the central nervous system. These neuroglia is like the glue of the neurons. Okay, they are all lumped together. They are all like a glue. It includes many types of cells, each with different functions. We'll see a little of these types of cells in a minute. And in the, what is PNS?
Peripheral
Yes, peripheral nervous system. And in the peripheral nervous system, this neuroglia is composed of cells that are called Schwann cells. Have you heard this name before?
Yes.
Probably vaguely you can remember the Schwann cells. And these Schwann cells form the myelin sheath, the covering. It's like a fat covering that insulates part of the axons around the nerve fibers. Okay. And also composed of satellite cells. All of them will protect and cushion the neurons. So in the central nervous system, we mentioned earlier, we have different types of neuroglia in the central nervous system. Astrocytes, microglia. Shapes are different. Astrocytes case, for example, they are abundant. There are a lot of them. They have this shape of stars, so they are star-shaped cells. They have numerous projections, embracing neurons, and they form a barrier between the capillaries and the neurons. They control the environment, the chemical environment in the brain. And the microglia here is like a spider. It's the shape of the spider. It's a type of phagocyte, meaning a cell that will dispose of dead cells and bacteria. Okay? Other type of neuroglia in the central nervous system are the ependymal cells. These are cells that are covering the central cavities of the brain and the spinal cord. Remember when we talk about the dorsal cavities, cranial cavity and the spinal cavity, these cells are lining these cavities. And they have some cilia, and when they move their cilia, they are helping the cerebrospinal fluid, this fluid, to move it. Okay? Helps to circulate the cerebrospinal fluid. We also have another type of neuroglia called the oligodendrocytes. These are important because in the central nervous system they produce the myelin sheath around the fibers of the nerves. Okay? So in case of the central nervous system, it's the oligodendrites producing the myelin sheath. And they can coil around different fibers at the same time. And this is a big difference with the neuroglia producing myelin sheath in the peripheral nervous system. Okay? So an asterisk here. Okay? Important fact. What's the difference between neuroglia and neurons? The glial cells do not transmit the nerve impulses. These are supporting cells. Okay? And the glial cells can divide. The neurons cannot. So if you start killing your neurons by doing drugs or you're exposed to toxics, then the number of your neurons will start decreasing. That is why most of the brain tumors, you know that tumors are usually overgrowth of the cells, but uncontrolled growth. Most of the brain tumors are gliomas. Okay? They are coming from glial cells growing uncontrolled. And what happens in the peripheral nervous system? Here we find the Schwann cells. Okay, these are the Schwann cells, and they will form this myelin sheath, this covering in the axons or in the fibers of the neurons in the peripheral nervous system. And a big difference with the oligodendrites in the central nervous system is that here the Schwann cells will wrap only one nerve fiber. Okay? We also have satellite cells in the peripheral nervous system that will protect the body of the cell, the body of the neuron. Okay, so if we have here a figure, this will be the satellite cells protecting the body, and each one of these is the myelin sheath produced by a Schwann cell. Okay. So basically, neurons are also called nerve cells. They are highly specialized. They will transmit nerve impulses. And what are some common characteristics to all these types of nerves? Basically, we did have the body and the process. This is the body. This part, the body and the process. Here and here. Okay? The processes are going away from the body. And the cell body will contain the nucleus. The cytoplasm surrounding the nucleus will have the centrioles. And if you remember maybe a little bit about division of cells, cell division, you remember the centrioles play some part in division of cells. So here we don't have centrioles because they're not going to divide. And this is the metabolic center of the cell, okay? Meaning that metabolic processes are happening here. Okay, so we have the cell body and we have the processes, okay? Do you remember the names of the processes? Of the nerves? We have the dendrites and the axon. Okay, these are the processes. And these are fibers extending from the cell body. Are you remembering a little bit from your classes in high school? Okay, what will happen if the body of this neuron is damaged? What do you think will happen to that neuron? The neuron will die. So it will not be replaced. Okay? And talking about the processes, we mentioned we have the dendrites here, these processes, and we have the axon, the long one. So the dendrites actually conduct impulses towards the cell body. Okay? So the impulse will come here, this way. These are the dendrites. And they may have hundreds of dendrites depending on the type of neurons. You may have hundreds of dendrites. And a big difference between the dendrites and the axon is that the axon conducts impulses away from the cell body. So information coming to the neuron will come through the dendrites, but information the impulse going out will go through the axon. Okay? It's like a one-wedge in this. So the axon will conduct the impulse away from the cell body, usually only one axon, and it ends in axonal terminals. These are the axonal terminals. Okay? What happens at these terminals? If we take a closer look, this is one neuron, and this is what we call postsynaptic neuron, but these would be another neuron. Okay? So the axonal terminals are separated from the next neuron by these space or these gap. Okay, they're not touching each other. there is a very tiny space between them called the synaptic cleft. And this junction, this area here, this junction between two neurons that are communicating is what we call synapse. Okay, there are many new words maybe, so try to remember. This is what we call synapse. So the neurons never touch other neurons. Okay. Are we good here so far? There's a lot of information maybe. So let's talk about the myelin sheath. This is an extended and modified plasma membrane. It's the plasma membrane of the oligodendrocytes or the Schwann cells that are wrapped. They are wrapped around the nerve axon. Okay. If you see closer to the figure, if we imagine that this yellow tube is the axon of a neuron, and then the light blue is the Schwann cell. So it's a Schwann cell plasma membrane that is wrapping around the axon. Okay, and what is the function of these myelin sheath? They will protect and insulate the fibers. They will increase the transmission rate of the impulses. We will see how in a minute. And we mentioned this. Who produces this myelin sheath? In the central nervous system, the oligodendrocytes. These oligodendrocytes can wrap many nerve fibers. And in the peripheral nervous system is the Schwann cells. But the Schwann cells only will wrap one nerve fiber. Okay? And here is a task for you. I have mentioned many times the oligodendrocyte and the Schwann cells and how they are wrapping the nerve fibers. Okay? So there is a difference between the nerves that are in the central nervous system and the peripheral nervous system. When we damage our nerves in the peripheral nervous system, they still can regenerate. Why? What is the connection between the regeneration of the fibers and the Schwann cells? Okay? Look for that as a homework.
Can you repeat?
Okay. What is the relationship between the Schwann cells and the fiber regeneration in the peripheral nervous system?
Regeneration of
Schwann cells and regeneration, fiber regeneration. Okay, so we mentioned that the myelin sheath also increase the transmission rate of the nerve impulses. If we have, this is one Schwann cell, this is another one, this is another one. Actually, between both of them, there is a space. And this space is called node of Ranvier. When the impulse is being conducted through these type of neurons that are myelinated because they have myelin, the transmission of the impulse will go from each node to each node of Ranvier. So it's like jumping from node to node. But if the neuron doesn't have the myelin sheath, then the impulse will have to travel along all the way of the axon. Okay, so imagine yourself, you have to go from here to there, and then I have to go step by step, each step. It will take me more time than if I go jumping From here, jump, jump, jump. So that is basically the idea why they increase the transmission. There are short portions of the axon that are not covered by myelin, and they are critical in propagation of nerve impulses. Now there are a few terms that we have to have very clear. So I put this terminology slide. Basically, in the central nervous system and the peripheral nervous system, how do we call when we have a group or a cluster of cell bodies? Okay. In the central nervous system, a cluster of cell bodies will be called a nucleus if it's a singular, or nuclei if they're plural. And the nerve fibers, the bundle, the group of nerve fibers, will be called tracts. This is just another way of calling them. But in the peripheral nervous system, this group or this cluster of cell bodies are called ganglion if it's one, or ganglia if it's more than one. And the bundles of the nerve fibers in the peripheral nervous system are just called nerves, the one we know, nerves. Okay? Okay, so probably you have seen many pictures or figures, slides from the brain, right? So in the central nervous system, we have white matter and gray matter, right? Why is it called white and gray? If you make a cut, you will see it darker or lighter. So basically, why is it darker or lighter? Because the white matter are composed of myelinated fibers, and the gray matter are the unmyelinated fibers and the cell bodies. So the gross macroscopy, if we see them with our eyes, we can actually distinguish this is gray and this is white. Okay? This is if we have this cut like this, frontal plane. We see this. And this is if we have a spinal cord cut transversal. Okay? If we see the function of the neurons, we can classify them in three types. One will be the sensory neurons, the one carrying the impulses from the receptors to the central nervous system. And usually, this will be the sensory. This is the sensory nerve, the sensory neuron. The cell bodies of these neurons are found in the peripheral nervous system. Okay? Usually here we'll have a group of them and we will call them ganglion. Okay, remember the terminology slide. Ganglion, a group of cell bodies. So information is coming here. This is our sensory cell. It's coming to our spinal cord. Then we have another one, our motor neuron, carrying the efferent transmission. The motor neuron will carry the impulse from the central nervous system, either to the visceral, stomach, intestines, so on, or to muscles and glands. Okay? And the cell bodies here in the motor neurons are in the central nervous system. So while this one is in the peripheral, it's out, this one is in the central nervous system. Okay? And then we have a third group called the interneurons, or association neurons, which is this one. They will make the connection between the sensory and the motor neurons. And of course, they are within the central nervous system, so the cell bodies are also in the central nervous system. But this is a classification according to their function, right? What function they do. The function. If we see the structure, then we can classify them as multipolar, bipolar, or unipolar. The multipolar has several processes. This is usually the classic figure of a neuron when we are studying the neurons. We have the cell body, the dendrites, the axon, and the axon terminals. So this is a multipolar neuron. It's the most common type. We find them in motor and association neurons. Remember from the previous one, in motor and association neurons. Okay? And the bipolar neuron will have an axon and a dendrite. But these type of neurons are actually rare. You can find them only in the eye or in the nose. So if I ask you to draw maybe or describe, to draw a neuron, this should be the figure, okay? The general idea of the neurons. And then we also have the unipolar neuron. Like this one, we have the cell body, a very short process. And an axon, a short single process, and the sensory neurons in the peripheral nervous system. But in this case, we have an axon going this way and this way. And transmission also goes, what I mentioned earlier was transmission goes dendrite towards the cell body, and in the axon goes away from the cell body. But not in this case. In this case, we'll go here and here. Okay? It goes both ways, towards and away from the cell body. So we mentioned earlier the top two functions of the neurons. Do you remember? Irritability and conductivity. What was irritability?
Respond to stimulus.
Yes. And conductivity? This is easy.
Send.
Yes. Transmit the impulse.
Transmit impulse.
Okay. So irritability, the ability to respond to the stimulus and convert it into an impulse. And conductivity, to transmit this impulse to other neurons, to muscles, or to glands. Okay? Very important, the two main functions of the neurons. So the next part is a little bit tricky. It's a little bit long explanation, so I will make the explanation and then we will watch a video. Maybe with the video you will have a clear idea in your heads. When the neuron is not transmitting impulse, when it's at rest, we say that the plasma membrane of the neurons is polarized. Okay? And this is related to the charge of the ions that are contained within and outside the neuron. So at rest, when it's not transmitting impulse, there are fewer positive ions inside than outside. Okay? There are less or fewer positive ions inside than outside. Then as long as the inside remains more negative, because we have fewer positive ions, so it's negative on the inside. So as long as the inside remains more negative than the outside, the neuron will be inactive. Okay, so it's at rest. The major positive ions we have inside are potassium. We have potassium ions. And on the outside, you have sodium. Okay? So what happens? This is at rest. It's polarized. So on the inside, there are more negative charges. And on the outside, positive charges. What predominates is potassium inside and sodium outside. Okay, you remember the figure with the glass and the water, and we were talking about solutions and solute and which way they move and so on. So now we'll have to remember that. So if a stimulus opens the channels of sodium in a certain part of the membrane, which is called a patch, then the sodium will go inside the neuron, right? The sodium will diffuse quickly inside. But the sodium has positive charge. And in the inside, we have mostly negative. So what happens with a lot of ion sodium is coming inside? It will change the charge of the inside, correct? Sodium coming inside, they will start becoming more positive. Okay, but only in these parts, right? Where the channels were open, we are starting to have a change in the charge. So it's starting to depolarize. Okay, depolarize. What kind of stimuli can trigger this? Diverse types of stimuli. It could be light, it could be stress, it could be a neurotransmitter, could be a sound or a pressure. But in the end, the result is the same. Okay? So whatever stimuli comes, these change in polarization will be the same. Okay? But in order to have an impulse to continue having these change in polarization, we need this stimulus to be strong enough, okay, and to have a great influx of sodium, a lot of amount of sodium coming inside, okay, enough that we can create an impulse, or what we call an action potential. Okay, the nerve impulse is called action potential. And one characteristic of the action potential is that it either happens or it doesn't happen. So there's no half action potential. That is why they say it's an all or none response. Okay, either happens or it doesn't happen. So basically the membrane will change its permeability. A lot of sodium will come inside. After the sodium rushed in, then the potassium has to rush out so that the neuron can restore the electrical condition. Has to go back to repolarization, has to turn around. A lot of sodium has come inside, now it's positive, but we have to put it back to a negative state. So in that case, because we have more potassium inside and it's positive, then we will make it go outside the neuron so that it's again negative charge inside. Okay? So we have a repolarization. Okay? At rest it was polarized, then it depolarized, then we repolarize. Okay? So the sodium-potassium pump will restore the original concentrations of sodium and potassium. Remember, this is for the electrical condition. But now we're having more potassium outside. But we need to bring potassium inside and to move the sodium outside because that's the normal state. So we will use for these sodium-potassium pumps. But this is an active process that will need ATP, okay, adenosine triphosphate. Remember when we were talking about active and passive movement. So if sodium does not enter, there is basically no action potential, okay? And until repolarization, until the membrane goes back to the original state, the neuron cannot conduct another impulse. Okay? The impulses will travel faster when the fibers have nearly sheath. We mentioned this. the impulse will leap, will jump from node to node, and this is called saltatory conduction. Okay? So if any of this explanation was not very clear, we will watch a 3-minute video. Pay attention. The URL for the video is not on the handouts, but it's on the slide, so take note once the video is finished. Okay? Let's go.
Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way.
Okay, and this is the URL if you want to take note and watch again the video. You can direct it from here. Okay. So it was clearer with the video maybe? You have to go home and reread again, okay? Okay, did you finish taking note? Can I go to the next one? So here's a question. How does the electrical impulse will get across the synapse to the next neuron? We mentioned one neuron and the next neuron, they're not touching, right? They're separated by this gap, this cleft, synapse cleft. And we are saying the impulse is traveling along the membrane, an electrical impulse, a change between the ions and it's coming here to the axon terminal. How does it move to the next neuron? Otherwise it goes nowhere. It will jump. Okay. That's an idea. Another one?
Axon terminals release neurotransmitter.
That's right. So pay attention. The question is how the electrical impulse will move to the other side. It doesn't. Okay, so the electrical impulse does not move to the next neuron. Okay, what you are now doing is transmitting this impulse and changing it using the neurotransmitter. So this is what was going to happen in the synapse. Remember, this is the area where one neuron is connecting with the next neuron. This is the synapse. The transmission at the synapse. The action potential will reach the axon terminal. So the action potential comes and reaches the axon terminal. The vesicles containing the neurotransmitters will fuse with the axonal membrane. So you have some neurotransmitter at the end, at the axon terminal. These vesicles will fuse with the membranes and they will release a neurotransmitter. This neurotransmitter will be released into this synapse cleft, into this space, and they will diffuse across and bind to receptors on the other side. So basically this is what happens. We have here the action potential, arrives to the axon terminal, and there you have these vesicles containing the neurotransmitters. Okay, these tiny vesicles will fuse with the membrane. This is your neuron. The vesicles are here. The membranes of the vesicles are fusing with the membrane of the axon terminal. Then the neurotransmitter is released into this synapse cleft. Neurotransmitter here. And on the other side, you will have a receptor for this specific transmitter. Okay, so once the transmitter binds to the receptor, it will open the channels for the sodium to come inside. So the action potentials is started in the dendrite of the next neuron. So these movement of the sodium coming inside will start the next action potential. Yes? And the neurotransmitter has to be removed. Otherwise, this gate will be permanently open to sodium coming inside, right? So the neurotransmitter has to be removed. It's quickly removed from the synapse, either diffusion, reuptake, or just breakdown. Then the neurotransmitter is removed, so the sodium stops coming inside. And again, you have start again your action potential. The neurotransmitter is released from a nerve's axon terminal. The dendrite of the next neuron has receptors that are stimulated by this specific neurotransmitter, and the action potential will start in the dendrite of the postsynaptic or the next neuron. The neurotransmitter is quickly removed by different means. Okay, so that is why we say the transmission of the impulse is an electrochemical event. The neurotransmitter is a chemical, right? And these change of positive and negative ions, sodium, potassium, and changing the polarization of the membranes, this is an electrical process. So the transmission of the impulse is electrochemical. Okay, so we will skip the part of the reflexes. If you want, you can read it. We will not touch on that. And next class, we will start from here, from the central nervous system. We don't have much time left. I don't want to cut after the first slide. So, questions, comments? No? You can go for this chapter to the same book that I suggested for the skeletal system. Okay? You can read it slowly, carefully, see again the figures, try to figure out the process. How the action potential starts and move along is very important part of the physiology in the nervous system. Okay, so this is important. Then for those of you who want to come on Friday, remember E-Building, seminar room, 4:30. Okay? Any questions? Not compulsory. You can come if you want, if you have questions. Okay?
Next week we will start at central
Yes.
nervous system.
Yes, I'm afraid last year I didn't have time to do reproductive system. I'm afraid maybe this year we may also not have time for reproductive system because it's not really. I would like to cover first the main systems.
As for the neurons,
Yes,
for the impulse,
yes,
do they jump from one knot to another,
Yes.
another knot? From the knot of
yes, yes, they do. Okay, if there's no more questions, I will see you either Friday or next Monday. Have a very nice week. If you have questions about the assignment, also please let me know, okay?
May I know the percentage breakdown of the 60%?
What do you mean?
The final exam is 40%, right?
There is final exam.
Yeah, so it's 40% of total.
I forgot how much, but yeah, I think so. Right?
So how is the breakdown of the another 60%?
I mentioned in the first class, do you have it?
Syllabus.
Yes, but I think in the handouts I put something different. Do you have the handout of the first class?
No.
No?
You
have?
You didn't say.
Oh, I didn't.
Did you say?
Oh, okay. Then we will go with the syllabus. Syllabus, how much it says? 40% final exam. Quizzes and assignments 60. Okay. The quizzes and assignments are together, 60%, right? But the quizzes, let me check very quickly. We have 2 minutes. Altogether, the quizzes and the assignment mean this topic? Yes, it's going to be 60%. But how much is for each of it? Assignment is 20. Yeah, assignment will be 20%. And the quizzes, 3 quizzes with your highest score will be 40. So, yes. If you have 3 good quizzes, it's like an exam, pretty much. The idea is for you to review constantly every week so you don't have a lot to take on the final exam. So if you work week after week, have good grades in the quiz, do a fairly good assignment, it shouldn't be that bad on the final exam. Okay? Oh, I didn't put this. There you go.